Biologists have historically debated how to best divide the massive field of biology, whether by major life groups like zoology and botany, by study levels ranging from molecular and cellular to organismal and population biology, or by form and function like anatomy and genetics. Yet, regardless of whether it is studied in a general biology or specialized molecular department, microbiology stands out as the ultimate key to understanding existence. Microorganisms are the most exceptionally attractive models for decoding fundamental life processes because, mind-blowingly, at the cellular level, these vital processes are performed in the exact same manner, whether they occur in a tiny microbe, a laboratory mouse, or a human being. They are an investigator's dream. They can be conveniently grown in compact test tubes and flasks, requiring vastly less space and maintenance than plants or animals. And they reproduce at an incredibly explosive rate, with some bacterial species undergoing nearly 100 generations in a single 24-hour period. Because of this rapid life cycle, scientists can observe them in microscopic detail while they are actively metabolizing, growing, reproducing, aging, and dying, and by simply modifying their environment, researchers can alter their metabolic activities or even change specific details of their genetic patterns without ever destroying the organism. The biological parallels are staggering. For instance, yeasts utilize glucose in essentially the same way as mammalian tissue cells, employing the exact same system of enzymes to break down sugars, trap the liberated energy, and make it available for cellular work, proving that the mechanism of energy utilization is fundamentally universal across the entire biological world. However, their energy sources are vastly more diverse. While plants rely strictly on radiant energy from the sun, and animals require chemical substances for fuel, some microbes act like plants, others like animals, and some possess the completely unique superpower to utilize both radiant and chemical energy, with certain bacteria able to consume an unbelievable variety of substances ranging from simple inorganic compounds to highly complex organic matter. This makes their physiological and biochemical potential greater than all other organisms on Earth combined. For example, bacteriophages, which are viruses that infect and reproduce inside bacteria, provide a perfect observable model of complete host-parasite reactions that helps us understand viral infections in higher plants and animals, proving to be of inestimable value for decoding complex genetics. Furthermore, while some bacteria can literally pull nitrogen straight out of the atmosphere to synthesize proteins, others require specific nitrogen compounds as building blocks. While some microbes can synthesize every single vitamin they need from scratch, others must have them provided. By organizing a vast collection of these organisms from the simplest to the most complex nutritional requirements, biochemists can actually map out the exact evolutionary pathways of cellular synthesis, tracking how life builds everything from atmospheric oxygen to complex amino acids. It is this astonishing versatility and fundamental connection to all living things that led the late Rutgers University professor Selman A. Waksman during his 1942 presidential address to what is now the American Society for Microbiology to declare that there is absolutely no field of human endeavor, whether it be in industry, agriculture, food preparation, clothing, or the conservation of health and combating of disease, where the microbe does not play a dominant role. And just 10 years later, in 1952, Waksman proved his own profound point by winning the Nobel Prize in Physiology or Medicine for his part in discovering the life-saving antibiotic streptomycin, which was, amazingly, produced by a simple soil bacterium. If you want to keep exploring the invisible forces that run our world, hit subscribe to join us at Microbio and comment below telling us what we should put under the microscope next.